Welcome. In this session, I'll be continuing discussing about the processes of rDNA technology. That is the steps involved in rDNA technology. So the steps involved in rDNA technology. So in the last session, I gave you the various steps involved in this rDNA technology, like isolation of the genetic material, then cutting the DNA at specific locations, then amplification of gene of interest using PCR. Okay, so I think we need to discuss from that step, that is the third step, amplification of gene of interest using PCR. Okay, now once we cut the DNA at specific locations by restriction endonucleases, so whatever that fragments of DNA we obtain, we will link those fragments to different plasmids. So one fragment will be attached to one plasmid and put this plasmid into a host cell like these bacterial cells and allow them to grow. And whatever that DNA that we have linked to the plasmids will start expressing once they are present inside the host cells, and thereby we can identify our gene of interest. So once we have identified the gene of interest, of course, you can make that into number of copies by using that gene cloning, okay, process, or by making a number of copies with a very short period of time. So within a short period of time also you can make a number of the gene of interest. So how is that possible is by using the PCR technique. So here the PCR stands for polymerase chain reaction. So this one stands for polymerase chain reaction. Okay. Now, in this polymerase chain reaction, so first let's see what are all the requirements, okay, of this PCR. So requirements. So it requires, okay, enzymes. So of course we know the PCR is just like DNA replication. So it requires enzymes. What that enzyme, the main enzyme that is required is a DNA polymerase enzyme. So DNA polymerase enzyme is required. But the point is that the normal DNA polymerase enzymes will get denatured, okay, when the temperatures are raised, because this PCR, the polymerase chain reaction, is performed in a machine, okay, called Thermocycler, in a machine called Thermocycler. And this Thermocycler will be able to, okay, raise the temperature and decrease the temperature automatically. So it will control the temperature. Now when the temperatures are raised to high, say suppose about 90 degrees Celsius, then what happens is this DNA polymerase, means normal DNA polymerases, will get denatured. So normal DNA polymerase enzymes denature at high temperatures. Denature at high temperatures. So for that reason, I cannot use the normal DNA polymerase enzymes. So scientists found, okay, that a bacterium named Dermus aquaticus. So this bacterium, Dermus aquaticus, lives in very hot environmental conditions, and this has enzyme DNA polymerase that is thermostable. So it has DNA polymerase enzyme that is thermostable. And that thermostable DNA polymerase, they have named it as Taq polymerase. They named it as Taq polymerase. In Taq polymerase, okay, so this T is the first letter of the genus and AQ are the first two letters of species. So it's called the Taq polymerase enzyme. They named it Taq polymerase, right? That is obtained from this bacterium called Dermis aquaticus. So this is a bacterium from which it is obtained. It is thermostable enzyme. So this Taq polymerase is thermostable, and it is no doubt it is a DNA polymerase enzyme, but it is a thermostable enzyme. So I'll just mention here, instead of the DNA polymerase, I'll mention here the Taq polymerase. Okay. So that is, of course, as I said, it is the DNA polymerase. Then next, it requires primers. So these primers are oligonucleotides. So which means a very much short stretch of DNA molecules will be seen, right? So which will be complementary to that fragment of DNA at the points. So primers are needed, and without primers the process of PCR cannot take place, because the primers will provide third OH ends, and we know that the polymerase enzymes will never start a new strand by their own. They compulsory require these third OH ends. Without that they won't start the process of extension or making a new strand, right? So it requires enzymes, the primers, and also it requires those nucleotides. It requires nucleotides. So these are the things that are required to perform this PCR. So now let's see the steps of this PCR. So there are 3 steps which are involved in PCR. So one, it's called denaturation, another one called annealing, and the third step is called extension. So each will be taking place at a different temperature. Okay, say this denaturation occurs at 94 degrees Celsius, whereas annealing occurs around 50 to 60 degrees Celsius, and then extension may take place at 70 degrees Celsius. And once this extension is completed, okay, again the machine will raise the temperature to 94 degrees Celsius, where the denaturation takes place. So within a short period of time, if you put one DNA fragment, it can make 1 billion copies in a very short period of time, right? So now let's see these steps, that is denaturation, annealing, and extension. So here the first step, the denaturation. So let's see that PCR. This is the gene of interest. Now when the temperature is raised to 94 degrees Celsius, the denaturation takes place. Okay, and what happens is, in the process of denaturation, the hydrogen bonds which are present will get broken down, thereby separating the two strands. So this separation of the two strands due to breakdown of hydrogen bonds is what we call the denaturation. So now in the next step, so once these two strands get separated, The next step will be annealing. Now the temperature, the thermocycler will automatically reduce the temperature to say 50 to 60 degrees Celsius. So it brings the temperature to 50 to 60 degrees Celsius, and now once these temperatures are reduced, it results in attachment of these primers. So I told you the primers are going to provide the third OH end. So this side it has 5 dash and this side it is 3 dash, and here it is 5 dash and this is 3 dash. So it means that the polarity of the strands, it will be 3 dash and 5 dash here, and this is 3 dash and 5 dash. So this attachment of primers to the DNA strands is called annealing process. Now in the next step, okay, that is called extension. So which takes place, now the temperature is slightly increased to 70 degrees Celsius. Already primers are attached. Okay, so now the DNA polymerase, that is here it is the tag polymerase, will attach on the third OH end, and it starts adding nucleotide by nucleotide, and then it goes on increasing. So this will become the new strand with the 5 dash to 3 dash, and earlier it was okay 3 dash to 5 dash. The same thing will happen here also. The tag polymerase will add on new nucleotides, and this will become 5 dash to 3 dash, and this is already 3 dash to 5 dash. So now you can see, once this extension is completed, the one DNA molecule became two DNA molecules. So I say, okay, this one DNA molecule, after first cycle it becomes 2, then second cycle it becomes 4, then the third cycle it becomes 8, okay, 16, 32, and goes on in a geometric fashion, right? And you can see after some around 30 cycles, so after around 30 cycles, you can see that the gene of interest is amplified to around 1 billion copies. Okay, so this gene of interest, after 30 cycles, approximately 1 billion copies are made. So there are 1 billion copies. Okay, so this is the process of PCR, which is used to amplify the gene of interest to a number of, okay, of course that is the amplification, which means increasing the number of copies, right? So that's all about the PCR, a very important one. So the next one, the fourth step, that will be insertion of rDNA into host cell or organism. So insertion of rDNA into host cell or organism. So I think that is what we have already studied in competent host, transformation with the rDNA. So we have used various methods to make the host cells to take up the rDNA. So I told you, once we prepare the rDNAs, okay, I'm going to take the host cells Okay, so this is a host cell, and you can see here I have taken the rDNA. All these are rDNAs. Right. So we can apply, different methods like what we have seen by treating with the divalent cation like calcium. Okay, then putting on ice, then giving a heat shock, and then again putting on ice. Right, that is one method that we usually use for the bacterial cells, and various methods for transformation. Now, after that methods, if these host cells take up the rDNAs, okay, if they are going to take up that rDNAs, okay, now these host cells who have taken the rDNA, as I told, they are called the transformants, and the one who did not take, these are the non-transformants. And of course, earlier I told you that how to identify the transformants and non-transformants. So by linking that gene of interest to antibiotic resistant genes. So when we have seen the plasmids, like in case of PBR, okay, 322, I told you that this is the region where there is a tetracycline resistant gene. So when you link, okay, a gene of interest to this tetracycline or ampicillin resistance, or whatever you can take. So this is ampicillin resistant gene, and if you're going to insert the alien DNA or the gene of interest here, okay, into this, it will result in changing that sequences of that gene. As a result, what happens is, so this is the gene of interest, and this results in the non-functionality of ampicillin resistant gene. So ampicillin resistant gene will be no more working, and this will actually help us to identify the transformant and non-transformant. So here all the transformants will be having non-functional ampicillin resistant gene, whereas the non-transformants, so I'm sorry, transformants will be having non-functional ampicillin resistant gene, whereas non-transformants will be having both ampicillin resistance and tetracycline resistant genes are functional. So the transformants will not be able to survive on ampicillin. So that's, I say, the transformants will not survive on ampicillin. Okay, so that indicates those are having, so when they die, we can come to know that they are having those rDNAs in them. So that's the insertion of rDNA into the host cell or organism here, right? So clear that one. Of course, we have studied that those ampicillin resistant genes and tetracycline resistant genes both are the selectable markers. So this ampicillin resistance and tetracycline resistance, these two are the selectable markers, means the one who will help us to select the transformants and non-transformants. Okay, then the final step in this rDNA technology is obtaining the foreign gene product. So here I have inserted this foreign gene into the plasmid, and I have put that plasmid into the host cell where it starts showing its expression. Say suppose this is the gene that actually produces insulin. Now this plasmid, this part of the plasmid which has my gene of interest will produce insulin. Now point is that I need to get this insulin in large quantities. We know the biotechnology is meant for the benefit of human beings, so not just one, for all the human beings. So hence I need to produce large quantities of this insulin. So for that purpose I need to grow these bacteria, okay, in a nutrient medium, okay, not just in a small petri dishes and all. I need to grow them in large containers, what I call them bioreactors. So let's see the last step of this rDNA technology, obtaining the foreign gene product. So the last step is obtaining the foreign, okay, gene product. Right. And one more point. So as I have taken the example of insulin, so here the bacteria, say suppose I'm using the bacteria as a host, okay, for this process now. Okay, so this is a host organism, and this host organism it doesn't produce insulin. So bacteria don't produce insulin. So it's in case of humans we produce insulin, and that gene which produces insulin I have now introduced into the bacteria and making this bacteria to produce that insulin. Now in such a case this bacteria is called heterologous host. It's called heterologous host. So a heterologous host is a host that normally doesn't produce that particular protein. Of course, insulin is a protein hormone, right? And it is not produced by this bacteria, anything. So if I keep a gene into one of my cell that doesn't produce that product, then that is said to be a heterologous, okay, host or cell. So in this case this bacteria is a heterologous host, okay, so where I have introduced the insulin, and that product what I obtained from this heterologous host is called the recombinant protein. So the product obtained from heterologous host is what I say the recombinant protein, and it's called the recombinant protein. Okay, I hope these two words are clear, the heterologous host and the recombinant protein. Now I told you that once we transform the host cells we need to grow them in large quantities so as to obtain that product in large quantity. So here what I have did, I have taken the plasmid, and to that plasmid I have linked the gene that codes for insulin. Okay, so this is, say suppose, the E. coli bacterium, okay, and this is rDNA, and that is having insulin gene, okay, and this is the genome of bacterium. Now if I allow this bacteria to divide, then what happens, the rDNA present in the E. coli will also start dividing. Okay, so for this, what are all the temperatures, pH, at what pH, at what temperature, and what are all other external factors that are required for this E. coli to grow at its faster rate. I'm going to supply it by using something called machines, that what we call the bioreactors. So these bioreactors are usually made up of some non-corrosive materials like this stainless steel and so they're made up of right, non-corrosive materials, they right. And so say I'm just I'll draw later about those bio the structures or types of bioreactors, we'll see it. So this is a bioreactor, and under this, okay, in the bioreactor I'm going to manage the temperature. So temperature, then pH, oxygen supply, and whatnot. Okay, everything I will be controlling. So a computer connected to it will control all those temperatures, even mixing the contents inside, so that the bacteria or the host organisms or cells will be able to get enough oxygen, and so that they multiply at their faster rate, so that we get that recombinant protein at the earliest, right. Right. So we'll see now those types of bioreactors. The most commonly used bioreactors are stirring type. So stirring type of bioreactors are used. Okay, they are large vessels. Okay, very big containers. Okay, and let's see, there are mainly two types we will see about those stirring type of bioreactors. One is a simple stir tank bioreactor, and another one called the sparged stir tank bioreactor. So these are simple stir tank bioreactor and sparged stir tank bioreactors. Okay, first we'll see the diagrams of these two, simple and sparged, and there later we'll see further explanation on them. Let's see, these are the two types of bioreactors, that is simple stir tank bioreactor and the sparged stir tank bioreactor. So here stirring is common in these two types of bioreactors. Okay, so I told you that the bioreactors will provide optimum growth conditions. So they provide optimum growth conditions. Okay, so they will have optimum temperature, so they will have optimum pH, means optimum, so whatever that maximum temperature at which the microbial activity can be maximum, so that is optimum temperature, pH. Okay, and then the optimum substrate that is provided in the reactor, and then okay, optimum salts, then vitamins, whatever that are required for the growth of these microbes inside the bioreactor, including the oxygen. Now, so here in case of simple stir tank bioreactor, so you see inside there is a flat bladed impeller. So this is bladed impeller, and this fat bladed impeller will be rotating because it is connected to a motor. So this will be rotating. Okay, and inside this is all filled with the medium, right? In that medium there will be the the transformants. Okay, so whatever that transformants which we have created, we will be introducing into this, and they will be okay growing at this optimum temperature, pH, whatnot, all by using these vitamins, oxygen. So they will be multiplying and increasing in the number, and at the same time the gene of interest that is present in the rDNA of those transformants will also get expressed, and they will be producing whatever that the gene of interest codes for. Okay, and that will be released into the medium, right? So now this simple stirred tank bioreactor, so it has flat bladed impeller that is connected to a motor which rotates here, and there is an okay inlet for acid or base, okay, for pH control is present here. So that is okay, so they are going to add acid or bases to maintain a particular pH inside the bioreactor. So because different host organisms or cells require different pHs, so by altering the acid or base, they can control the pH. Then here is an inlet for steam, that is for sterilization. Steam for sterilization. Okay, so to remove unwanted microbes that are not required, because whenever there is a nutrient medium, it will attract many other microbial organisms to grow on them, not just that intended organism that we would like to grow in this bioreactor, right? And this simple stirred tank bioreactor also has okay a foam breaker. It also has a foam breaker. So while okay mixing the contents inside, it results in the formation of foam, and that foam will be removed by this foam breaker. So that's what we see here in case of simple stirred tank bioreactor. Of course, it is all filled with the culture broth. Is filled with the culture broth. Okay, and coming to the sparge stirred tank bioreactor, in this okay we see there is air bubbles. So sparging is passing that air bubbles through the liquid medium, right? So these are air bubbles, and this is the culture broth. So you can see the air bubbles coming okay from down, and here this will be rotating here, so that the contents are mixed well, and these air bubbles actually increase the surface area. So this is the one rotating in case of a sparge stirred tank bioreactor. Of course, this will be also connected to a motor that will rotate at a particular speed. Okay, so that will be also control everything. So all these will be actually connected to kind of their own computer systems, which will help in maintaining. They automatically regulate by detecting the pH. They release acid, okay, or base, and try to maintain the pH. And same thing in case of this sparge stirred tank bioreactor also, right? Now a bioreactor, so any of these bioreactors, they have something called agitator system. They have agitator system. So this the flat bladed impeller which is connected to the motor, and it rotates and mixes the contents inside, is agitator system. So agitation is mixing, right? And then it will also have oxygen delivery system. So it will also have oxygen delivery system. So it will be providing oxygen into it. Of course, we need to see whether these are aerobic or anaerobic. So in this case, we are considering aerobic. So it means there must be supply of oxygen, okay, into this culture broth. So that oxygen should be uniformly distributed all over. Otherwise, if the oxygen is not available, it can result in some other reactions or death of that host cell or microorganism, and that results in the complete failure. So there must be oxygen delivery system attached to these bioreactors. Then there is a foam control system. There is foam control system. You can see here, this is what I said, the foam breaker. So that is a foam control system. So it removes that foam, okay, formed when there is a process of agitation. Then there is temperature control system. So because we know that enzymes are mostly affected by the temperature, and it's very important to maintain that optimum temperature. So without that, okay, we will be not able to get what we are, okay, have done all this for. Then they also have the pH control system. Also have that pH control system. So okay, already that I have discussed. Then it will also have the sampling pots. We'll also have the sampling pots, pots. So they have the sampling pots, okay. So these sampling pots will be withdrawing small volumes of culture to see that how far the culture is going good, whether it has been with some other microorganisms are growing along with that, or whether the reaction is going all well. All that we can come to know that by using these sampling pots, from which we can withdraw a small amount of the sample and we can test, okay, so how it's going on here, okay. Then so in the sparse steel tank bioreactor, so here is increased, so this area is increased surface area, okay. So that's an increased surface area, of course, for that oxygen transfer or oxygen transfer. So that's all about this simple steel tank bioreactor and sparged steel tank bioreactor. Now once we grow those microbes, or what are the host cells or organisms in the bioreactors, okay, say suppose as I have taken the example of insulin, so I have grown the bacteria that have the the rDNA, which consists of this insulin gene, and that insulin gene got expressed and it has produced insulin, which actually released into this culture broth. So it means that the insulin is mixed with this, okay, culture broth. And now what we need to do is we need to separate that insulin from the culture broth, purify that, okay, then add some preservatives and whatever that formulations required, and then make them into bottles and then release into the market. So all this process, what we call it, the downstreaming, right, process. So let's look at that downstreaming process. So the last step is downstream processing, right? So the last step is downstream processing. So let's see the definition of this downstream processing. Okay, so the process of separation and purification of the product. The process of separation and purification of the product is called the downstream processing. And this downstream processing has some steps. Okay, so one, it will be the first step will be the separation and purification. So I told you that whatever the product that is produced by those transformants will be released into the culture broth, and we need to separate that product from the culture broth, purify it, okay, and then add some other chemicals, okay, that will be under formulations. And once it is done, so we need to release into the market. Okay, so under that, the first step will be the separation and purification. Then formulation with the suitable preservatives. Formulation with the suitable preservatives. So means we need to add some preservatives, okay, that can keep those products or increase their lifetime. Okay, so formulation with the suitable preservatives. Then we need to do even clinical trials. So clinical trials in case of drugs. So if there are medicines, like for example this insulin, this has to be this need to undergo clinical trials, means they are going to test on some animals first. Okay, so step by step on mice, then on monkeys, and then later they will try, okay, on few human beings and see that whether it's working or not. So if it is not working, then they need to go and again see what's the problem, and then again repeat that whole process, okay, again. So they need to go for these clinical trials, okay, if it is in the case of drugs. And then the last one, there must be right a strict quality. So we need to maintain that quality, so there must be strict quality control. Must be strict quality control testing should be done. So these are all the steps which we see under this downstream processing here. Right, so once this is finished, now it can be released in fact into the market. So this is right all about the biotechnology principles and processes. With this we have completed this chapter. So in the next session we'll see about the next chapter, biotechnology and its applications.